Hello everyone, I am Tejaswini of second sem microbiology. Today I am here to present seminar on the topic Bio-Remediation of Soil. What is bioremediation? Bioremediation is the combination of two words. Bio means living and remediate means to solve a problem. By definition, bioremediation is the process of cleaning up environmental sites contaminated with chemical pollutants using living organisms, which transforms hazardous components into less or non-toxic substances. Sometimes it essentially removes the contaminants from the soil or the area which are polluted. The process of bioremediation is typically carried out by microbes like bacteria and fungi. Sometimes bioremediation process is also associated with plant interaction. This technique is also termed as phytoremediation. Bioremediation technology using microbes was reportedly invented by George M. Robinson. As I mentioned, the contaminants may be naturally occurring compounds in the environment that are present in unnaturally high concentration, example crude oil, phosphates, heavy metals, etc. Sometimes the contaminants may be chemically synthesized compounds that are never occurred naturally, example xenobiotics like pesticides, hydrocarbons. Is there any necessity to remediate soil? Let's understand. Soil is a vital mixture of minerals and organic materials, liquids, moisture that supports the growth of a rich variety of plants and microorganisms. It is the supportive terrestrial ecosystem of our planet. For all its wonderful properties, soil also easily gets contaminated by pollutants which are released either intentionally or unintentionally. Many anthropogenic activities like industries, disposal, mining, application of fertilizer, etc., or unintentional process such as oil spills, fuel dumping, etc., causes pollution in the soil. Since soil is such a complex mixture of minerals and organic materials, it is not trivial to remove contaminants. These contaminants can end up sticking to other chemical compounds in the soil, or they can move through the soil along with rainfall, which ends up in sticking to the surface of waterways or underground water reservoirs. The biggest concern is associated with the harm it causes to the human health. Hence, there is obvious necessity to clean up the contaminated soil, and it is the one scientific way or the scientific method which is applied to restore the natural pollution-free state of the soil. Microbes involved in bioremediation process. Since I mentioned microbes are applied to remove the contaminants in the soil, let us see how the actual process goes on. During bioremediation, microbes utilize chemical contaminants in the soil as their energy source, and through oxidation reduction reactions, they metabolize the target contaminants into usable energy for other microbes also. The byproducts released back into the environment are typically in a less toxic form than the parent contaminants. Therefore, let us discuss which are the microbes are useful in the process of bioremediation. Bacteria in bioremediation. Aerobic bacteria such as Pseudomonas butida, Rhodococcus species and Mycobacterium species show the high level degradation of pesticides and hydrocarbon pollutants in the soil. Since these organisms are able to utilize the contaminants as their sole source of carbon and energy. Next, methanotropes. These are also aerobic bacteria that utilizes methane for their carbon and energy source. Methane monooxygenase is the enzyme which are present in these organisms, which has broad substrate range, active against a wide range of compounds such as chlorinated aliphatics. Anaerobic bacteria. These are not frequently used as aerobes but often applied to bioremediate polychlorinated biphenyls, which are simply called as PCBs, in river sediments, and a facultative anaerobic bacteria, Dichloromonas aromatica, can able to oxidize aromatic pollutants like benzoate, toluene, and this species is especially used for in situ bioremediation. Next, Deinococcus radiodurans. It is an extremophile which is resistant to radiation. It is used in bioremediation of heavy metal and mercury in soil. Alkanivorox borcumensis. It is a marine bacteria which consumes hydrocarbons and produce carbon dioxide. Next, fungi in bioremediation. Using fungi as potential treatment of contaminants began in the year 1985 when Phanerochaete chrysosporium, a white rot species, was discovered to metabolize multiple key environmental pollutants. Application of fungi in bioremediation is also termed as mycoremediation. White rot fungi has high ability to transform and mineralize specific pesticides, whereas brown rot fungi such as Formitopsis pinicola and Glyophyllum trabeum show an ability to eliminate DDT in soil. Archaea such as Halobacterium, Halopharax, Halococcus genus were well known to evaluate their potential to biodegrade crude oil and hydrocarbon in soil. Archaeoglobus fulgidus, an hyperthermophile, can break down various aromatic compounds. Phytoremediation, which refers to the technique which relies on the use of plant interactions in polluted sites to mitigate the toxic pollutants. Example, Serrantia marcensis, which is used to bioremediate gasoline contaminated soil, Z-mace, heavy metal contaminated sites. Requirements for bioremediation. The primary ingredients for bioremediation process are the presence of contaminants which are used by microorganisms as their energy source. Next, an electron acceptor to carry out the process of oxidation and reduction reactions, and presence of microbes that are capable to degrade specific contaminants. Essential factors for microbial bioremediation. Since microbes possess some limits of tolerance for particular environmental conditions such as temperature, pH, as well as optimal conditions for their effective performances, let us understand which are the factors that are going to affect the rate of microbial bioremediation. First, nutrient availability. Nitrogen, phosphorus, sulfur, etc. are some of the nutrients which are necessary for microbial activity and cell growth. It has been shown that treating petroleum contaminated soil with nitrogen can increase cell growth rate and decrease the microbial lag phase, and it also helps to maintain microbial population at high activity level along with increase in hydrocarbon degradation. It is also shown that excessive amount of nitrogen in soil cause microbial inhibition. Below 1800 mg nitrogen per kg of H2O is optimal for petroleum hydrocarbon bioremediation. Next, moisture. All soil microbes require moisture for their growth and function. Availability of water affects diffusion of water and soluble nutrients in and out of microbial cell. Excess of moisture is undesirable because it reduces the oxygen availability for aerobic respiration. 45 to 50% of water holding capacity is optimal for petroleum hydrocarbon degradation. Soil pH. It is one of the important factor because most microbial species can survive only within a certain range of pH. Furthermore, soil pH can affect availability of nutrients. pH 7 is optimal for biodegradation of petroleum hydrocarbon. Temperature. It influences rate of bioremediation by controlling rate of enzymatic reactions within microorganisms. Generally, speed of enzymatic reactions in the cell approximately doubles for each 10 degree Celsius rise in temperature. Most bacteria found in soil, including many bacteria that able to degrade petroleum hydrocarbon, are mesophiles, whose optimum temperature ranges 25 degrees Celsius to 45 degrees Celsius. Thermophilic bacteria, which are normally found in hot springs and compost heap, exist in cool soil environments and can be activated to degrade hydrocarbons with an increase in temperature to 60 degrees Celsius. Contaminants. Contaminants can adsorb to soil particles, rendering some contaminants unavailable to microorganisms as their energy source, which is crucial for biodegradation. In some circumstances, bioavailability of contaminants depends not only on the nature of contaminant but also on soil type. This is a slide which shows the reaction in aerobic biodegradation and anaerobic biodegradation. In case of aerobic bioremediation, the electrons in the contaminants are oxidized while oxygen gains electrons and is reduced. This results in the formation of carbon dioxide and water. In case of anaerobic, where the oxygen is limited, the inorganic compounds such as nitrates, sulfates, carbon dioxides serves as terminal electron acceptor to facilitate biodegradation process. Here the slide shows the sequential reactions which are taking place during bioremediation process, which depends on the nature of the contaminant. Types of bioremediation. There are two types of bioremediation broadly: in situ bioremediation and ex situ bioremediation. In situ bioremediation, where the remediation process is done at the site. Excavation of soil or the polluted site is not required. Treatment of contaminated materials or contaminated sites is done at the contaminated place itself. The major techniques which are used under this type is bioventing, biosparging, bioaugmentation, natural attenuation, biostimulation. Ex situ bioremediation. This process is carried out away from the contaminated site. The technique involves physical removal of contaminated materials for treatment process. The techniques under this type of bioremediations are biopiling, composting, bioreactors, and landforming. This slide shows the types of bioremediation according to the technique used. In situ bioremediation, which means there is no need to excavate or remove soil in order to accomplish remediation. It is superior method to cleaning contaminated environments since it is cheaper and uses harmless microbial organisms to degrade the chemicals. This technique involves the supplying of oxygen and nutrition by circulating aqueous solution through naturally occurring bacteria to degrade organic compounds. In situ bioremediation is applied to the degradation of contaminants in saturated soils and groundwater. There are types of in situ bioremediation: intrinsic in situ bioremediation and engineered in situ bioremediation. Intrinsic in situ bioremediation involves the conversion of environmental pollutants into the harmless forms through innate capabilities of naturally occurring microbial population. Engineered in situ bioremediation. It is the one which accelerates the degradation process by enhancing the physiochemical conditions to encourage the growth of microorganisms. Bioventing, bioslurping, biosparging, bioaugmentation, and biostimulation are few of the techniques used in engineered in situ bioremediation. Intrinsic attenuation. This is the method which relies on natural conditions and behavior of soil microbes that are indigenous microbial population of that contaminated soil. The use of natural process to control spread of contamination from chemical pollutants at contaminated sites. This intrinsic attenuation is also termed as natural attenuation. Bioventing, the process in which oxygen and nutrients are pumped through injection wells into the soil. This bioventing process is well suited for drained medium or coarse textured soil, whereas the bioventing process is prevented by clay soil since they are fine textured and possess smaller pores, high surface area to retain water. Bioslurping. It is a technique which is designed for free products recovery such as light non-aqueous phase liquids and to remediate soil contaminated with volatile and semi-volatile organic compounds. This technique is a combination of vacuum enhanced pumping, soil vapor extraction and bioventing by indirect provision of oxygen. Biosparging. It is the technique which is very similar to bioventing, but here the injection of air into soil is not done on the subsurface. Instead, here the injection of air or oxygen into the soil is done at the saturated zone, which causes upward movement of volatile organic compounds to unsaturated zone. Thus results bioremediation. Biosparging technique is widely used in treating aquifers contaminated with petroleum products, especially diesel and kerosene products. Biostimulation. It is the method which utilizes indigenous microbial population of contaminated site. In this technique, it consists of adding nutrients and other substances to the soil to catalyze the natural attenuation of the indigenous microbial population. Here, the nutrients such as nitrogen, phosphorus, sulfur, calcium, magnesium, etc. are added to stimulate the microbial population that are capable to remove the contaminants from the soil. Bioaugmentation. This involves the introduction of exogenic microorganisms which are capable to detoxify the contaminants. Sometimes genetically altered microbes are also employed in bioaugmentation process. Ex situ bioremediation. This is the technique that those applied to soil which has been removed from contaminated site via excavation or pumping, then it is treated elsewhere. Under this, solid phase bioremediation. Biopiling. Excavated soils are mixed with soil amendments and placed on the treatment area, whereas these biopiles are aerated with use of perforated pipes and blowers to control oxygen supply. This biopiling system is used to remediate oil and hydrocarbon contaminated sites. Next, land farming. It is one of the simplest technique due to its low cost and less equipment requirement for the operation. Excavated polluted soils are carefully applied on fixed layer which is supported above the ground surface, and these are allowed aerobic biodegradation of pollutants by native microbes. Composting. It involves the combination of contaminated soil with non-hazardous organic amendments such as manure and agricultural wastes. Under slurry phase system, this is one of the rapid process compared to other treatment process. The contaminated soil is combined with water and other additives in a large tank called bioreactors. Bioreactor, as the name implies, it is a vessel in which raw materials are converted to specific product or products following series of biological reactions. There are different operating modes under bioreactors such as batch, fed batch system, continuous or multi-stage. All this depends on contaminant and economy. For example, stir tank bioreactors are used for crude oil polluted sediment. Submerged bioreactors are used to remove effluents, whereas packed bed reactors are used to bioremediate amine pollutants. Advantages of bioremediation. Bioremediation, it is a natural process and therefore perceived by the public, and bioremediation is used for the complete destruction of a wide variety of contaminants. This process can prove less expensive than other technologies that are used for cleanup of hazardous waste. Bioremediation can often be carried out on site, often without causing a major disruption of target pollutants is possible. Instead of transferring contaminants from one medium to another, that is, for example, from land to water or water to air, the complete destruction of target pollutants is possible by the technique of bioremediation. Next, disadvantages. There are some of the disadvantages of bioremediation, and they are: bioremediation is limited to those compounds that are biodegradable. Not all compounds are susceptible to rapid and complete degradation. There are some concerns that the products of bioremediation may be more persistent or toxic than parent compound, and it is difficult to deduce from bench and pilot scale studies to full scale field operation. Biological processes are often highly specific. Microbial populations, suitable environmental growth conditions, and appropriate levels of nutrients and contaminants, all this matters when we come across bioremediation. Bioremediation often takes longer time than other treatment options. These are the references for this particular topic, bioremediation of soil.